First of all, I'm super excited to welcome Dr. Kristen Zuloaga, which I probably said wrong, to today's talk. I'm very, very excited about her presentation, but before we get to it, I should do a land acknowledgement. I want to recognize that many of us work, live, play, and participate in a community on the unceded, ceded, and traditional territories. This is in Canada, so I would look it up for your own country, of course. Of the 203 First Nations, along with the 38 Métis chartered communities, each of which possess their own unique traditions and history here on this land that we now refer to as British Columbia. That's where I am. We acknowledge the importance of the Truth and Reconciliation Committee of Canada's call to action, the United Nations Declaration on the Rights of Indigenous Peoples, and the BC Declaration on the Rights of Indigenous Peoples Act. In all of the work, we're committed to ensuring Indigenous women's rights to health and safety and equal opportunity to participate in a manner that recognizes and respects Indigenous cultures and traditions. Again, I really emphasize that it's really important to look in your own communities at the Indigenous cultures that you are living in. Today, I'm joining you from North Vancouver, which is part of the unceded homelands of the Coast Salish peoples and the traditional territories of the Musqueam, Squamish, and Tsleil-Waututh First Nations. Probably many of you are well aware, but this is part of the Women's Health Research Cluster Seminar Series. We have over 300 members worldwide. We run a number of different events. There's some of them that are restricted to UBC, like that Tri-Cluster Research Day, I think is restricted to UBC, but most of our events are not restricted at all. We have another workshop coming up on how to gender. That's not what we called it, but I'm super excited about that one too. So look for announcements on that. It's sometime in November. We couldn't do this without support from people. You can support us too if you have a little bit of loose change around. So take a look at our website. And we also have a number of travel awards. I know people aren't really traveling yet, but there'll be a new round of awards coming up for trainees in November. So also take a look for that, all of you trainees. We have very, very good success rates. So please do think about it. And anyone can join, anyone can be a member. So we don't limit that. Now I'm going to stop sharing my screen. Where am I to stop sharing my screen? I did it the wrong way around. And now Kristen can start sharing her screen. And while she's doing that, I'm going to read out her bio. So Kristen is an Assistant Professor of Neuroscience at the Albany Medical College. She obtained her PhD in Biomedical Sciences in 2011 and has been trained by leaders in the field of neuroendocrinology, cerebral vascular physiology, and cerebral ischemia, aging, and neurogenesis. And I could read out all the names, but I won't in the interest of time. She serves on as both a council member and education co-chair for the Organization for the Study of Sex Differences. I know that because I'm president-elect of that same society, and it's an amazing society, and Kristen does an amazing job on all of those committees as well. It's true. I'm not just saying that. She also serves on the Editorial Board for the Biology of Sex Differences, and if you don't know about that journal, a very good one to check out. She's deeply committed to mentorship. I also concur with this, and is a co-founder, co-director of the Albany Postdoctoral Development Program. And now, as you also might know, I like to talk about a fun fact about the presenter. And Kristen's fun fact is that one of her lab's favorite traditions is dressing up for Halloween. I actually love dressing up for Halloween myself as well. Sometimes I do it as a witch, and my husband always says, that's not really a good costume. It's a joke, you know. Anyway, and they do reverse trick-or-treating. So they hand out candy to other labs, admin staff, and our animal caretakers. I love that. And now I have to go to Albany just to go and get some candy. So without further ado, Kristen.
Thank you so much. And thank you so much for the invitation. I am super happy to be here. I've attended you guys' some of your seminars in the past, and they're wonderful. So thank you for offering them to others as well. So today I'm going to be talking about our work looking at sex differences in metabolic and hormonal risk factors for dementia. So our lab studies several different types of dementia. So obviously Alzheimer's disease, you've all heard of. This is the most common form of dementia, and it's due to buildup of toxic proteins in the brain, such as amyloid beta and tau neurofibrillary tangles. You also get a lot of neuroinflammation as well, and brain atrophy. The second most common cause of dementia is called vascular contributions to cognitive impairment and dementia, or VCID. And this is another major focus of our lab as well. And this is dementia that's caused by either a reduction in blood flow to the brain. So you can think about if you have carotid artery stenosis, maybe you're not getting enough blood flow to the brain, or damage to the blood vessels within the brain. And this can be from many different causes. One of them is, interestingly, from amyloid beta protein building up in the blood vessels, and this can cause microbleeds and ruptures. So there is some overlap between these two diseases. Also, if you have vascular dysfunction, this can also impair the clearance of amyloid as well. So there's this reciprocal relationship between the two different forms of dementia. And so with that, then, it's probably not surprising that there's often a lot of overlap between the two. And so when you have both pathologies present at the same time, this is called multi-etiology dementia. And so if we look at the population of patients that have Alzheimer's disease, it's been shown that about 70% of these actually have multi-etiology dementia, an overlap between this VCID, or this vascular form of dementia, and the Alzheimer's disease pathology. And although this is very common clinically, this is not something that's very well studied in our animal models. And so one of the things that we're doing in our lab is incorporating these vascular risk factors as well into our animal models. So I'm really excited about risk factors. I think that they're a great thing to be targeting in dementia, because there's these studies that have come out that have shown that a lot of dementia could either be prevented or significantly delayed just by targeting risk factors. So about 40% of dementia cases they think could be prevented or delayed just by targeting these 12 common modifiable risk factors. And I've highlighted 3 of these here in purple because they're ones that we're particularly interested in, because they're related to metabolic health. So if we think of obesity, of diabetes, of physical inactivity, which puts you at risk for these metabolic disease, these are things that all make this list of these top 12 things that we can do to modify our dementia risk. And so looking at metabolic disease, it's very interesting because it presents as a risk factor in midlife. Now, you don't normally get a dementia diagnosis until late life, but at midlife, this is actually when the pathology begins to build up in the brain. So in your 30s, 40s, and 50s, there's already changes that are happening in your brain, which are going to later lead to dementia. And it's during this time in midlife when metabolic diseases such as obesity and diabetes are going to increase your risk of dementia. And so if we look at the case of obesity, this is going to increase risk for both Alzheimer's disease and VCID, but it's going to be a stronger risk factor for this vascular form of dementia. For diabetes, this is going to double your all-cause risk of dementia, but there's a sex difference here in the context of VCID or vascular dementia, where diabetic women are at increased risk compared to diabetic men. Metabolic disease is extremely common in dementia patients, particularly in Alzheimer's disease. So about 80% of those with Alzheimer's disease have either prediabetes or type 2 diabetes. Now, this could partially be because the metabolic disease is a risk factor in itself, but it's also because this Alzheimer's pathology can have metabolic consequences as well. Another risk factor that we are very interested in is biological sex as a risk factor. So about 2/3 of all those with dementia are women. And this is, of course, partially because women live longer than men. So with this longer lifespan and with aging being the greatest risk factor for dementia, it's probably not surprising then that more women present with dementia, but this doesn't fully explain the sex difference. And so one of the reasons it's thought that women might be at greater risk is that they might have more exposure to risk factors throughout their lifespan. And so there is a female-specific risk factor that we've become interested in. This is a midlife hormonal risk factor, and that's menopause. So about 20% of the US population right now is postmenopausal women. I apologize for not looking up Canadian statistics, but I'm guessing it's pretty similar in Canada as well. So this is a large portion of the population. Almost any woman who has dementia is going to be postmenopausal because it is a disease of aging. And menopause itself has been linked to white matter loss. It's been linked to reduced brain glucose metabolism. So there's some changes in the brain that occur during menopause that are going to put you at increased risk for dementia. And then, so it's probably not surprising that the earlier that you go through menopause, the higher dementia risk you're going to have. And so our lab is focused on these dementia risk factors and kind of the intersection of them. So we're interested in these metabolic risk factors like obesity and prediabetes, vascular risk factors, like if we have a reduction in blood flow to our brain or blood-brain barrier is leaky, and then also sex differences. So we're interested in differences between males and females, but then also these sex-specific endocrine aging that take place. So in females, this would be menopause, and males, it would be andropause, and kind of looking at how all these come together. And so I want to go over briefly how we model some of these metabolic and hormonal risk factors and these different types of dementia in mice. So one of the models that we're using that's the newest for us is our menopause model. And we were looking for a model that would be as close to the hormonal profile that women go through during menopause as possible. And here I'm focusing on just one of the hormones, estrogen, because this is the hormone that has been shown to have neuroprotective effects. It is anti-inflammatory. It increases blood flow to your brain. It has all these very, very wonderful benefits for your brain. And so it's something that is also lost with menopause. And so if you are premenopausal, but after puberty, in general, you have high estrogen levels. These are going to be fluctuating with the menstrual cycle, but in general, they're higher. After menopause, they're very low, but there's also this time period in between of perimenopause where you get these huge fluctuations in hormone levels. And women experience these during perimenopause as vasomotor symptoms such as hot flashes. And this time period can last several years in women. And so this is something we're interested in mimicking. Now, what's normally done in rodent models, which is what we're using, is a surgical menopause model. And we use this in our lab as well for some of our studies. And this is where you remove the ovaries, which are the source of these hormones, and you get this immediate sharp decrease in the hormone levels. This is great because it is fast. It is going to be very reproducible between all of your animals as far as the timeline. But we didn't like the fact that it's missing this perimenopausal phase. And so we were looking for another model. And there was a model that was developed by Pat Hoyer's lab. And we were working with Hedwin Brooks to develop this model in our lab. And it's an accelerated ovarian failure model. And so this model uses a reproductive toxin to induce ovarian failure. And what's really great about it is that the hormonal profile that the rodents go through is very similar to the hormonal profile that women are going to go through. And so they have these high estrogen levels. Then they have a perimenopausal phase, which is going to last several months, which is, equivalent roughly to the several years that it would be in women if you're trying to do the math between mouse and human. And then they go into this low estrogen state. And so I'll go into a little bit more detail later about how this works, but this is something that we were very interested in looking at this model that would also include this perimenopausal phase. And so one of the reasons we were interested in mimicking this phase is this is when hot flashes happen. And in these mouse models, people have even been able to measure hot flashes with their skin temperature changes. And hot flashes in women are linked to more white matter hyperintensity, which you can see on an MRI. And so these white matter hyperintensities are normally indicative of vascular damage that has occurred. And so we think that this might be something that's very physiologically relevant to mimic this perimenopausal phase. So that's how we're modeling menopause, and how we're modeling metabolic disease is a model that's very commonly used. It's a chronic high-fat diet. The diet we're using is not a healthy diet. It's not ketogenic. It's not Mediterranean. It is 60% fat from lard, still has 20% carbohydrates. The mice become obese and they become pre-diabetic. depending on the timing of when you initiate it, if you do it very soon after weaning, males can actually become type 2 diabetic, but the age we're doing it, they're going to be pre-diabetic. And so this high-fat diet has been shown to cause vascular pathology, even specifically pathology in cerebral vessels. It impairs memory, and this memory impairment is reversible. So if we take a mouse that's on a high-fat diet and you put them back on a low-fat control diet for a month, you can actually see some memory impairments. You can even see a little bit of an improvement in blood flow. But these previous studies were done, including my own, in single-sex studies. And so this is what I was doing when I was a postdoc. And so when I started my own lab, I wanted to look into sex differences. And so I'll be sharing some of that with you today. For our dementia models, our vascular dementia, or VCID model, is a model of chronic cerebral hyperperfusion, meaning we are going to be reducing blood flow to the brain. And so how we do this is we go in and we isolate one of the carotid arteries. And so we're going to tie that off. We're going to cut off blood flow through one of the carotids. And then what this is going to do is it's going to cause a reduction in blood flow to that side of the brain. It's not a stroke. So because you have this circle of Willis where these vessels are interconnected, you're still going to be getting blood flow to that area. It's just going to be reduced. So you don't get an ischemic infarct or anything like this. Immediately after the surgery, which you can see on the right, and you see more of this blue-green color. And so this is a reduction in blood flow on the right side of the brain. This is with laser speckle contrast imaging. Immediately after the surgery, so 5 minutes after, we get about a 45% decrease in blood flow, but this quickly recovers over time. And 4 months later, it's around 15%. So we've done this with laser speckle contrast imaging, and also I did it with arterial spin labeling MRI when I was a postdoc. And so this is a persistent low level of hyperperfusion that then causes cognitive deficits. For our Alzheimer's disease model, we have 3 different models we're using in our lab right now. I'm going to introduce this one because this is the one that I'll be showing data for today. It's the one we've been using the longest. And so it's a triple transgenic model. And so this model is going to be making more amyloid beta. The type of amyloid they're making is going to be more toxic, and it also has tau mutations. And so these mice, the studies I'll be showing you, they're going to be at an age, an early middle age, when they are already going to have cognitive deficits, they have neuroinflammation, they have amyloid beta pathology, they have hyperphosphorylated tau at that age, but they don't develop plaques until a little bit later. Or sorry, tangles. And then finally, the last model I want to talk about is our multi-etiology dementia model. And so in this case, we'll take an Alzheimer's disease mouse model, and we'll perform this VCID surgery in those mice to reduce blood flow to the brain, to have both that vascular and that amyloid pathology in the same model. And so using these different models, we wanted to know if there are sex differences in the effect of midlife metabolic disease. And so one of the first things we were looking at is just simple metabolic changes. Normally, if you give a high-fat diet in mice right after weaning, which is what most people do, so they do this, you know, when they're still juveniles, the males have a much greater weight gain than the females. So we put our male mice on a high-fat diet. It didn't matter if they got a sham surgery or if they got the VCID surgery to reduce blood flow to the brain, they're gaining a lot of weight. However, when we did this in the females, we were very surprised to see that they're gaining much more weight. And so this is because they were given the diet at middle age. We started at 8 months, or sorry, we started them at 8 months of age. And so these females are actually gaining more weight than the males. We did a study where we compared different ages of onset. And so if you start it in juvenile mice, males gain more weight. If you do it in young adulthood, so around 2 months of age, they gain equal amounts. And then if you do it in middle age, now the females have this susceptibility. So along with this greater weight gain in the high-fat fed females, we also saw greater amounts of glucose intolerance. So this is an indicator of prediabetes. We saw more visceral fat accumulation, and the visceral fat is the pro-inflammatory bad fat. So this is the type of fat that's going to have more negative health consequences. And we've also done this same study in our Alzheimer's disease and multi-etiology dementia. I'm not showing you the data here, but we did see, again, the same sex differences in the Alzheimer's disease and multi-etiology dementia mice as well. So we've looked at some of the effects of this high-fat diet on memory. And what we found using a novel object recognition test to look at episodic memory is that we do see impairment with a high-fat diet. So in this test, mice are exposed to 2 objects. An hour later, we'll remove one of the objects that's familiar and replace it with a novel object. And we want to see that they're spending more than half of their time with this new object, indicating they remember the old one. And so when we do this in our male mice, whether they got this sham surgery or the VCID surgery to reduce blood flow to the brain, we do see that this high-fat diet is causing an impairment in episodic memory. We didn't see an impairment with just the reduction in blood flow to the brain alone in the males. When we did this in the middle-aged females, at the end of the study, when we're doing behavior, they're now 15 months of age, so they've been on the diet since they were 8 months old. We now see that the females are also having this impairment with the high-fat diet that we observed in the males, but also just reducing blood flow to the brain, and these middle-aged females also cause an impairment in their episodic memory. We did this as well in the multi-etiology dementia study, and we see with multi-etiology dementia, there's complete impairment in episodic memory in both sexes. It's a little bit worse in the multi-etiology dementia compared to the Alzheimer's disease mice, but not significantly so. One of the other things we wanted to look at was a measure of spatial learning and memory. And so we did the Morris water maze. There's 3 different parts to this test. So the first part is non-spatial memory. So we have the mice are in a kiddie pool of water that's been made opaque with non-toxic white tempera paint. And there's a little platform that has a flag on it that they're trained to swim to. And so over the course of 5 trials, they will learn how to swim to this little flag and get to the platform. Then we'll give them 24 hours to rest and recover. And then we take the flag off of the platform, and now they have to navigate to a hidden platform that's below the surface of the water using spatial cues around the room. And this is a test of spatial learning. And so then again, we'll be looking at their path length and how efficiently they're able to get to this platform. 24 hours later, again, after they've had some time to rest, they will then have what's called a probe trial, where we're looking for their spatial memory. In this case, the platform is removed altogether, and we're looking to see what % of time they're spending in that target quadrant where that platform would have been. And so when we did these trials, we don't see any differences in the visible trials, the non-spatial memory in any of our dementia models. We also didn't see any differences in the spatial learning component, so the hidden trials in our VCID model. When we looked at the male mice in the project, we were comparing wild type Alzheimer's disease multi-etiology dementia mice, regardless of whether they were on a low-fat diet in the solid bars or a high-fat diet in the hash bars, we also didn't see any effects. However, when we looked at the female mice, we saw that as we layered on more pathology, we did start to see some impairments in their spatial learning. So the Alzheimer's disease females that were on the high-fat diet had a longer path length in order to get to this platform. And then the multi-etiology dementia females, regardless of which diet they were on, also showed impairment in this spatial learning. So now we have females that are more susceptible to the spatial learning deficit than the males were. We also looked at spatial memory. So here I'm showing probe trial results from the VCID projects for reducing blood flow to the brain. In this case, we actually do see an effect of this surgery in the males. It didn't matter which diet they got. If we reduce blood flow to the brain, they are having an impairment in their spatial memory. When I had previously done study using arterial spin labeling MRI to look at changes in blood flow, you can look in deeper brain structures. And we do see with this surgery in male mice that there is a reduction in blood flow in the hippocampus. And so this is a more hippocampal dependent task than the episodic memory test that I had showed previously. And so the fact that we see a deficit with the surgery in the male mice makes a lot of sense, because we know that this is a brain area where the blood flow has been reduced. And then when we looked at the females, we saw a little bit of a different pattern. So in the females, the more pathology we layered on, we kind of saw this stepwise decrease in their spatial memory. So the females that got the sham surgery that are on the high-fat diet, they're not doing quite as well as low-fat mice, but they're still doing really good. There's not a significant difference there. If they got the VCID surgery, though, they are now showing this significantly impaired memory compared to our control females. And then if they got this surgery to reduce blood flow to the brain and they were on the high fat diet, now they're most impaired and they're just at the level of chance. We also have done this now in our multi-etiology dementia model. I'm not showing the data here, but basically spatial memory is impaired in both sexes with multi-etiology dementia. This was, we couldn't even look at differences in diet because we're just at floor effects because they were so impaired when we layered on these two dementias together. So one of the other tests that we looked at is nest building, which is a measure of activities of daily living. So this is something that is impaired in dementia patients. And to try to look at this in mice, you can think of dementia patients not being able to take care of themselves. And so in mice, what's commonly done is looking at their ability to build a nice nest for themselves. So in everything else I've showed you up until this point, our mice have been group housed for all of our tests because it's less stressful for them that way. For this one test, they are single housed overnight because we want to see how each individual mouse can build a nest for themselves. They are given 2 squares of nestlet, and then overnight we see how well they can build their nest. And so this is a really good nest if they score a 5. They have nice high walls. It looks nice and cozy. We have 3 people that are giving them a score that are all blinded to the treatment group, and then we will take the average of that score for this test. So when we did this in the VCID project, we actually didn't see any differences between our groups and their ability to perform this test. And when we did this in the Alzheimer's disease and multi-etiology dementia males, we also did not see any differences. They were all building pretty good nests. They were coming in at about a 4. So they have this nice nest. The walls could be a little higher. They were a little bit older at this point in time. So we'll cut them some slack for not being perfect. And then the females, though, we saw if they were in these Alzheimer's disease or these multi-etiology dementia groups, that they were actually not building nests. They were ripping up the nestlet, but they weren't forming these nice nests that the males were. So they were showing some impairment in this measure of activities of daily living. So we wanted to see if any of our cognitive measures and any of our metabolic measures correlated with each other. And one of the things that stood out to us across these studies is that the metabolic measures in the females were correlating with their spatial memory. So in females, the more visceral fat that they accumulated, the worse their spatial memory was. However, we didn't see this association in the males. And it wasn't just their amount of visceral fat, it was also their amount of weight they gained in general. And then we looked at some pro-inflammatory mediators in the visceral fat, because the visceral fat is supposed to be the more pro-inflammatory fat. And we found that TNF alpha, which is a pro-inflammatory cytokine in the visceral fat, was also associated with spatial memory in the females, but not in the males. We did these same associations in the multi-etiology dementia study, and we found this same sex difference again. And so next we wanted to look at some sex differences in the underlying neuropathology to see if that could be explaining any of these behavioral differences we saw, where the females seem to be more susceptible to having cognitive impairment. So the first thing we looked at was blood flow. We didn't really see anything too exciting here. If we did the surgery, it reduced blood flow to the brain. It was a little bit more severe in the females on the high fat diet when we looked immediately after the surgery, so 5 minutes later. But when we looked long term, there was no differences between males and females. no difference whether or not you were on a high fat or a low fat diet. And in general, there was about a 10 to 15% reduction in blood flow long term. Here's just showing that in the Alzheimer's disease versus the multi-etiology dementia mice, we see this decrease, but there's no differences. So that didn't seem to explain why we would be seeing any of these differences in cognitive function. So we also wanted to look at white matter pathology. So in VCID and vascular forms of dementia, white matter pathology is the main pathology that you observe. And so we wanted to look at some changes in white matter. So we looked at myelin through myelin basic protein, and we found that there was a main effect of surgery. So if the animals had the surgery, in general, they had less white matter than if they didn't have the surgery. But we also found an interesting sex by diet interaction, where the females that were on the high fat diet had less white matter than the males. And then we also looked at the correlations between their metabolic function and some of these white matter markers. And so we looked again at this myelin basic protein to see how it correlated with weight gain. No association in the males, but again in the females, the more weight they're gaining, then the less myelin coverage they have. We also saw this same association, not shown here, with visceral fat, with glucose intolerance. So basically these metabolic measures in the females are correlating with their loss of white matter. We looked at this in another way as well. So we looked at qPCR in the striatum, which is a white matter rich region. And we looked at OLIG2, which is a oligodendrocyte marker. And again, we found this association where the females who gained more weight had less white matter, and the males, we don't see this association. We also looked in the Alzheimer's disease and multi-etiology dementia project at amyloid beta pathology. We couldn't look at this in the VCID model because they don't develop it. And what we found is that our Alzheimer's disease multi-etiology females had much more amyloid accumulation than the males did. This has also been reported by other labs as well. What we also found is that there was a main effect of the mixed dementia. So the mixed dementia mice had more amyloid compared to just the Alzheimer's disease mice alone. But we didn't see any effects of the high-fat diet on amyloid accumulation. This is looking at amyloid positive cells. We didn't actually see this. Was earlier on the pathology, so they haven't developed severe plaques yet. There are some groups that have found effects of the high-fat diet to increase plaque formation. So if we had looked later on, we might have seen that, but in this earlier time point, we don't see that. And we didn't see any effects of sex or diet on tau pathology either. One of the other things that we looked at was neurogenesis to see, so in dementia, you are losing neurons. So wouldn't it be great if we could replace those neurons? And so we wanted to look in the hippocampus, which is an area important for learning and memory, and also an area where there's adult neurogenesis, to see if this diet was causing any changes in the new neurons that were being born. And so what we found is that the females that were on the high-fat diet actually had less new neurons that were being born. So the yellow-green color are double quartin positive new neurons. And we also found that when they were on the control diet, we did see that the females were having more of these new neurons being born than the males, but then the high-fat diet is then impairing that. And this is has been reported by others as well. And then we also looked at how the metabolic measures were correlating. And we found that, again, with the females, the more weight they gained, then the less neurogenesis that they had. And we don't see this association in the males. So the last thing that we looked at was neuroinflammation. And so we looked in a few different brain areas. The first one we looked at was the hypothalamus. So this is the metabolic control center of the brain. And since we're seeing these effects of high-fat diet and we're seeing this greater weight gain in the females compared to the males, we wanted to look to see if there was any changes in the hypothalamus. And we did see that the astrogliosis was much more severe in the high-fat fed females compared to the males in our Alzheimer's disease model. And this astrogliosis, again, correlated with weight gain in the females, but not in the males. And in the multi-etiology dementia mice, we also saw that astrogliosis in another brain region, the hippocampus, which is more involved in learning and memory, was also more severe in the females. And we also looked at microgliosis in that region. And so we found, again, with the females, that their metabolic measures are correlating. So in this case, we're looking at their glucose intolerance, the area under the curve in a glucose intolerance test. And the more glucose intolerant they have, then the more microglia coverage that they had. And so overall, what we found when we were looking at sex differences is that this high-fat diet was causing greater metabolic impairment in the females when we're initiating the diet in middle age, or if it's in one of these dementia models, and that there was a wider array of cognitive functions that were impaired by the high-fat diet in the females compared to the males. And we saw this in all the different dementia models we looked at. And that the high-fat diet was associated with reduced neurogenesis, more inflammation, and more white matter loss in the females. And that our multi-etiology dementia model, beyond just the Alzheimer's disease alone, did exacerbate the astrogliosis and the amyloid pathology in these mice. And we were excited because our findings were in line with clinical data that I mentioned at the very beginning that showed that diabetes is a greater risk factor for dementia, particularly VCID in women, than it is in men. And our model is not a type 2 diabetes model. It is a pre-diabetes model with obesity. And so we thought that this perhaps suggested that obesity and pre-diabetes might also be related to cognitive impairment in multiple forms of dementia in females. And interestingly, a study just came out a couple months ago, which was looking at prediabetes in the human population and did find that it was associated with earlier cognitive impairment and onset of dementia in women, but not in men. And so this is, again, kind of gives us some confidence that the animal studies are at least lining up with the human studies. And so now our lab's focus has shifted somewhat. We have shifted more to thinking about why females might be more susceptible to these negative effects of this midlife metabolic disease. And so we're looking at the role of menopause. And so we're doing this in basically all the different models that I've just told you about. So we are doing it in the context of just normal aging and the mild cognitive impairment that occurs from metabolic disease. And this is being led by Abby Solinero, my graduate student. We're doing it in the context of VCID, and this is led by Olivia Gannon, my other graduate student, and she has a fellowship specifically to look at this. And then we're also doing it in the context of Alzheimer's disease and multi-etiology dementia, and this is led by my postdoc, Dr. Charlie Abigano. So I mentioned that menopause at an earlier age has some negative consequences. So it increases your risk for diabetes, increases your risk for obesity, and particularly that central obesity, which is supposed to have more of these negative health consequences, and then also your risk for dementia and for cognitive impairment. And I mentioned before also that estrogen has all these protective effects. So it's anti-inflammatory. It also protects against multiple dementia risk factors. It promotes blood flow in your brain. It promotes your metabolic health, and your ovaries stop producing in after menopause. And so this is why one of the reasons that we're really interested in looking at menopause. So I mentioned we're using this accelerated ovarian failure model so that we can try to mimic this perimenopausal phase. And so I'll just briefly show a diagram of how this works. So this reproductive toxin is going to go in and it interacts with c-KIT receptors on the primordial follicles in the ovaries. And then this is going to cause atresia, cell death of these follicles. And what ends up happening is if you do histology on this ovarian tissue, you can see these follicles in the premenopausal mice, but in our postmenopausal mice, we no longer see any of these follicles. And so this is something very clear that we can validate the model with postmortem, but we also want to be looking to see when they're going into menopause. And so we track this with vaginal cytology, which is something that's very commonly used. So in women, they have their menstrual cycle they go through. In mice, they have an estrous cycle, which typically is about 4 to 5 days long. They will cycle through these 4 stages. But the menopausal mice get stuck in this diestrus phase. And so after they've been stuck in diestrus for 10 days, then we would consider them menopausal. And so one of the first things we looked at with this model is variations in the time that it takes to go into menopause. So the mice get injected with this VCD, this reproductive toxin, for 20 days. And then we look at how long it actually took them to go into menopause. And so it's around 90 days, but there is a decent amount of variability in this. This was with wild type C57 mice. We've also done it in a couple other mouse strains, and we do see some strain variation as well. So this is data in the APP NLF mice, which is another Alzheimer's disease mouse model. We found when they're on a control diet that they are actually taking a little bit longer to go into menopause than our wild type mice had. When they're on the high fat diet, they're actually going into menopause a little bit faster. We haven't seen that acceleration with the high fat diet in our wild type mice, but we do see in this Alzheimer's disease mouse model. So I think if you're interested in using this model, it's probably pretty important to validate it in your specific strain that you're going to be using so you can know what your timeline is going to be like. So we wanted to look at metabolic effects of menopause. So we know in women, it causes weight gain, it causes glucose intolerance, and other people had looked at this in other mouse strains. And so we have now done this in our C57 mice. This is in Olivia's VCID project. So the VCID part's not important for this part, but the menopausal mice, which are in purple, are gaining weight. They're not becoming obese. That's a moderate amount of weight gain, as you would expect. This is also more in line with what you would see clinically, and they are developing some glucose intolerance as well, which is a indicator of prediabetes. So Charlie has also done this in our Alzheimer's disease mice on both the low fat and high fat diet. And so if you see here in the black dotted lines, this is a mouse that got the control oil injections, and the purple dotted line is the mouse that got the VCD injections to induce menopause that's on a high fat diet. And you can see that the weight gain is accelerated with this menopause model, both in if they're on a normal diet, but even more so if they're on a high fat diet. And we do see that the glucose intolerance, which is shown on the right with the glucose tolerance curve, is a little bit worse as well in the menopausal mice. So it is accelerating their metabolic disease. So we're interested now in looking at cognitive effects of menopause. And so this is the newer part that we've been doing. And so far what Olivia has found is that the longer the mice are in menopause, the worse their spatial memory is. So because we do have variation in how long it takes them to go into menopause, we're able to take advantage of that variation to look at mice that are all the same chronological age. So the chronological age is the same, but what's different here is the endocrine age, so the amount of time that they've been in menopause. And so even though the mice are the same age, just this variation in how long it took them to go into menopause is affecting their cognitive function. So we're obviously interested, we haven't done any of this yet, in looking in the brains to see how this changes pathology. So in the Alzheimer's disease mice, does this change amyloid beta pathology? In the VCID model, is it causing white matter damage, vascular damage? In all the models, how is it affecting neuroinflammation? And then we're going to be using a brain-specific estrogen replacement therapy to try to see if we can alleviate some of this pathology and improve cognitive function. So those are all of the projects on the left are all the ones that I told you about where we're looking at modifiable risk factors. So metabolic risk factors, endocrine risk factors, which we can change with hormone therapy. But our lab is also interested now in looking at non-modifiable risk factors. So we have a new project where we're looking at genetic risk factors and specifically looking for Alzheimer's disease risk genes that have a vascular function, and seeing how those are contributing in the different sexes and with and without this metabolic disease. And I have zero data for that because it just started, but I'm excited about it in the future. And that is it. This is my lab. For not last Halloween, we have no group lab photo, but we are doing it this year. We're going to be puzzle pieces with our lab logo, and we're going to be adorable, but I don't have pictures yet. But this was from 2018 and 2019. You can see we're handing out candy, and it's super fun, and I love my lab. And that is it. Thank you to my lab, my collaborators, and our funding. And if you guys are interested in our work, please follow us on Twitter, visit our lab website, and I'll be happy to take any questions.
Okay, thank you so much, Dr. Zuloaga. I wrote it down phonetically. That was fantastic. If you can give some emojis and or unmute yourself and clap if you'd like to. Really, really exciting and fascinating. I tried not to tweet out any of the unpublished data, but I did do a lot of tweeting. So if you want to contact Kristen, you can do that by just following those tweets. I think you'll be able to see that.
Thank you.
Yeah, it's so super fascinating. I have a few questions, but I'm wondering if anybody else has any on the calls, because it shouldn't just be about my questions. And I hear that I think I've said this before, but if you wait a long time, there will be eventually some questions. But, I'm very type A, so it's hard for me to wait this long time.
I'm terrible waiting.
Hey, Stephanie. Yay. And Victoria. Perfect. Go ahead.
You go first, Steph. I was going to say, I'll let Victoria go first.
Oh, no.
I ask questions all the time.
Oh.
Thank you. Go ahead, Victoria.
Okay, Victoria.
Hi, that was an excellent talk. I have a couple questions with regard to your use of the VCD model. I'm from the Beaumonte Nelson Laboratory at Arizona State University, and we have used the VCD model in our rats. And I was looking at some of the body weight data, and I just had a question because I'm not as familiar with using VCD in mice. Do they reflect the same weight loss that's typically found in rats when you administer VCD? So do they have weight loss when you're administering it?
So we had one batch of VCD where they were having weight loss when we administered it, and we haven't ever had that happen again. So I think it was like something to do with like the batch of the drug, but we didn't have that happen like after the fact. So, and we had a cutoff. If they lost more than 10% of weight, we excluded them.
Can I ask
from
a
the
follow-up
study.
question that might be related? Is there a difference in the ages of, I know your rats, Victoria, and Kristen, your mice.
Yeah, and I think,
if,
what's your vehicle that you're using?
We are using DMSO in saline, so it's a saline.
Okay, yeah, we're using oil, so
okay,
maybe that might be why.
Yeah, we have used oil and actually found some differing results.
Yeah.
We're administering it IP then?
Yeah.
Oh, okay. And then are you doing it on like a 15-day injection paradigm or the 30-day?
Okay, so all the data that I showed, we were doing it for 20 days. We also have another project where we cut it off at 11 days because our mice it was an aged mice and they weren't doing as well on it. So we cut it down for them and we're only doing 11 and they still went into menopause. It just took longer.
Okay.
So.
That's interesting. Yeah, for our rats, we typically see weight loss as a result of across the 15 injections for VCD.
Yeah.
But there's a lot that plays into that. So it might be vehicle, it might be the IP injection that we administer, and also their age. We typically do it in older rats.
So I think that was also the first batch that we did was in our older animals. So we assumed it was because of the batch because it hasn't happened again. But to your point, it was also when we were doing it in the older animals and we've been doing it in younger ones now. So it might be it could be an age thing as well. But yeah, we just cut back on the amount of doses that we gave and they seemed fine after that. Like, so basically when they started losing weight, we just stopped injecting them and they still went into menopause. So some labs only use a 10-day protocol. So I think there's just variation. The more days of injections you give, then the shorter the timeline is going to be of how long it takes them to go in. So
I see. And so you're giving.
Can we if we don't mind, I'll just move on just because we have some more questions. Victoria.
Yeah, I have tons of questions for you, but I will stop.
Yeah, if you want to email me, I'd be happy to chat and compare experiences too.
Yeah, I love that.
We have one from Twitter that I really am invested in the answer, so I need to make sure we have time to ask that one. But Stephanie, you first.
Thanks, Lisa. And I'm like Victoria, I was trained in the Monty-Nelson lab, and now I'm postdocing elsewhere. But so excited to see people using the VCD model. But my question, I may have just missed this, is for your correlations with the body fat and spatial memory specific in females, were those adults or were they middle-aged mice?
So the VCID project, they were 15 months old when we were doing the behavior. For the Alzheimer's disease and multi-etiology dementia mice, they were in the 7 to 8 month old range. If we had waited until they were older, we would have had even worse floor effects with our behavior just because of how it progresses. So we had to do it earlier for them. So basically we had to change the timeline in order to not do that. We're now doing studies in a different Alzheimer's disease mouse model that doesn't get pathology, doesn't get cognitive impairment until 18 months of age, so that we can actually induce the menopause at a later age.
A little further. Yeah, that's interesting. I was just thinking if they were adults, which is, they're older adults heading towards middle age, if you had done it later or earlier, would you see the same effects? And then another quick question, if I may, for the high fat diet work, do the mice get any exercise? Like, do they have I know some people put wheels in the cages, or do they
ours do not. I would definitely expect that exercise would have protective effects. So it's going to increase neurogenesis. It would decrease their body weight. Like I would assume that it would definitely have protective effects. It's something I'd be interested in doing, but I would assume that would provide protection. But no, they're just in a normal cage. They are group housed, so they have playmates. They have friends to play with.
Yeah, no, I was just wondering if maybe like exercise might modify if they have the risk factor of the Alzheimer's model as well.
No,
as
it's a wonder
diet, how exercise might impact that. So anyway, really cool work. Thank you so much for
trying to
answer questions.
Oh wait, you're muted.
Gosh, I'm such a terrible mistake to make at this point in the pandemic. The question from the internet is, so this was a DM, okay? So just the question is, what do I eat? Low carb, high fat? What is a menopausal woman supposed to eat? And is there any evidence that there are some protective effects of, because I thought there were some, and I think in some of your outcomes, it wasn't like significant probably, but it looked almost like the high fat diet was almost protective in that.
So I think that, different high fat diets can have protective effects. So I think that if you were going to eat a high fat diet, that it was a Mediterranean diet, maybe even a ketogenic diet. So when women go through menopause, they kind of shift their fuel source to more ketone bodies. And so that might be protective, but like this particular diet, which is equivalent, I should have said this, I don't think I did. It's equivalent to eating double bacon cheeseburgers. So that's like the nutritional equivalent that they're matching. So basically that fast food diet, probably not the best choice, but I think that, I think
ketogenic.
I'm not saying all fat, like all high fat diets are bad. I'm saying like this high fat diet is bad, and we are using this high fat diet to model the obesity and the prediabetes that is so prevalent in our society today. But I definitely think that the formulation probably matters a lot.
The other thing that was really interesting to me, and then I'm going to leave the call a little bit early, but I know you have another meeting too. If Victoria wants to ask some more questions, she can, or other people, please put up your hand. The increased weight gain by the females, that's something that we're starting to look at some of this too, and we saw the same thing. So I'm wondering, but in rats, not in mice. I'm just wondering, like, what with the high fat diet, which is interesting. Like, you'd think that it must be palatable to both sexes. I don't know if people have looked at that, whether it's palatability, are they eating the same? We
didn't see differences in food intake, but we looked at it in a very like crude manner. So we were just measuring like how much food was left in the hopper each week in their group house. So we were, dividing it by the number of mice in the cage. So it was not like the best way to look at it, but we did not see any differences in their food intake per body weight. But I think that like more of what we're thinking is that it's those changes that are taking place in the hypothalamus that's then regulating like their feeding circuitry. So
interesting. sex
Yeah.
differences everywhere.
I know.
And I'm really pleased to see, you know, going from the sex differences to single sex, it's totally fine, right? I
Yeah.
did a little tweet about that, about how it's important to still, you know, yeah, the sex differences leads you somewhere. It's still important to follow up in a single sex study to figure out what that might be.
Yeah.
So with that, I apologize. I'm going to go off the call, but maybe Victoria, or is there anybody else that has any other questions first before I leave? Again, I'll talk really slowly. You know, sometimes it works, sometimes it doesn't work. Victoria, you can come off and ask a couple more questions, but I know that Kristen has another appointment too, so.